So first I'll start with Dr. Razak. He is a professor of psychology and the director of the graduate neuroscience program. He received his bachelor's degree in electronics and communications engineering from Chennai, India, from Anna University in Chennai, India. He received a PhD in zoology, physiology, and neuroscience from the University of Wyoming, where he also held a postdoctoral position along with a second position at Georgia State University. As you'll learn tonight, his research really focuses on auditory processing and brain plasticity using animal models to understand these human conditions. He has different lines of work looking at the neural mechanisms of sound localization and echolocation in bats, neural mechanisms of sensory processing abnormalities that you find in neurodevelopmental disorders like Fragile X syndrome, which is on the autism spectrum disorders, as well as how the auditory system changes with age and hearing loss, which we'll hear mostly about tonight. His work is heavily funded by the NIH, the Department of Defense, as well as an NSF Career Award. So I will turn it over to Khalil so he can tell us about his work on age-related changes in central auditory processing, mechanisms, and therapeutics.
Okay, thank you, Lani. Thank you, Aaron and Lani, for inviting me to this event and also helping me connect with the folks here in the webinar through the UCR Palm Desert Center. So thank you, all of you. So let me share my screen. Can everybody see my screen? Okay. All right, so as Lani mentioned, I'm in the Department of Psychology and also in the Graduate Neuroscience program at UC Riverside. The research in my lab is interested in understanding how our auditory system processes the everyday behaviorally relevant sounds that we hear and how such processing in the auditory system might change during development and during aging and with disorders which have communication implications, such as hearing loss and autism spectrum disorders. I focus mostly on animal models to understand the cellular and brain circuit level changes in auditory processing and also allow us to test and discover therapeutic approaches that might alleviate some of these changes with either aging or because of autism spectrum disorders. The main research themes in my lab currently are on why children and animal models of autism spectrum disorders have sensory hypersensitivity, and we test multiple transgenic mouse models for that purpose. The second major line of research, which I will talk about today, is on the impact of aging and hearing loss on auditory processing. Again, we use mouse models of aging and hearing loss to make these assessments. And another line of research that is ongoing in my lab for many years now is trying to understand fundamental mechanisms of hearing, and we study different bat models for that purpose. And I particularly study the pallid bat, which many of you might be from the southwestern desert might know it's a bat that is found here in our region. All right, so I'm going to focus on impact of aging and hearing loss today. So why study age-related hearing impairments? I should start off by providing some statistics on the prevalence of the condition. So for humans older than 65 years, approximately a third of us have hearing impairments that are either age-related or associated conditions that lead to hearing impairment. And for those of us who are older than 75 years, it's closer to 50% of people with hearing-related impairments. Hearing loss is typically diagnosed with an audiometric detection threshold test. It's also becoming widely recognized now that there's a condition called hidden hearing loss, in which the audiometric detection thresholds are pretty normal. So the threshold of hearing is normal, but central processing might be deficient. So both of these conditions are present with the direct hearing loss as well as the hidden hearing loss. And one of the major challenges for the auditory neuroscience field is to try to understand the relative contribution of hearing loss as well as hidden hearing loss to how the auditory system functions as we get older. And you might wonder, why is it really important to diagnose and treat age-related hearing loss? So I want to provide a paper. I want to inform about a paper that came out in 2017 This was initiated by the journal Lancet, and they got together a group of scientists to do an analysis of the existing literature to understand what are the major risk factors in developing dementia. Okay, so the paper was titled Dementia Intervention, Prevention, and Care, came out in 2017 with Livingston and others as the main authors. So here's one of the major figures that's highly influential in our field and many people are paying attention to. Basically, what this is showing you are the various risk factors at different stages of life, so either early life or midlife or late life, that are considered to be major risk factors in leading to dementia and cognitive decline. And typically, one would, if I were to ask you to guess what those factors might be, people would rightly claim smoking is one issue, hypertension, obesity, and so on and so forth. But what was surprising about this analysis was what you can see here is the largest midlife risk factor that leads to cognitive decline and dementia is hearing loss. 9% of the risk factor, which is higher than 2% hypertension and the obesity as risk factors. So this was a very important sort of discovery of existing literature, and they concluded that even mild hearing loss at middle age increases the long-term risk of cognitive decline and dementia. And after the publication of this Lancet Commission review, there have been a major interest in doing a number of studies asking, is this link present? And what is the reason for this link between hearing loss and cognitive decline? And greater than about 80 papers have been published in the last 5 years since the Lancet paper came out. And they mostly support the idea of hearing loss contributing to changes in quality of life, social relationships, motor skills, and function and morphology of specific brain regions, with the suggested idea that hearing loss leads to, or hearing loss as well as hidden hearing loss, so central processing decline, both of those combined can lead to speech processing defects, which can then lead to social isolation, which can then be mediating cognitive decline and dementia. So hearing loss is not just a sensory issue, but also seems to have broader implications in terms of cognitive function as we get older. These are just a couple of papers that highlight the specific relationships. One of the papers that came out, this is an older paper that was present in that review from Lin et al. They showed that for every 10 decibel increase in hearing loss, so once you reach a 25 dB threshold of hearing loss, every 10 dB above that, there was a 20% increased risk of developing dementia. And adults older than 65 years, the mean time for developing dementia was 10.3 years in those with hearing loss at baseline versus 11.9 for counterparts with normal hearing with a lot of other factors controlled. So this link is being established in multiple papers, and more papers have come out in the last 5 years on that. The causality of this link is controversial at this point, and that's why we need a lot of research into the mechanisms and understanding the relationships at the level of the brain itself in how this might be mediated. Some people have suggested that both of these might arise because of common factors, such as diabetes, vascular conditions may be common factors that lead to both hearing loss as well as dementia, and hearing loss itself is not causing dementia. So those are all the sort of the ideas that are being debated in the field right now. It's also likely that these associations are underestimates because in many cases where cognitive function is being tested, sensory factors are not taken into account as widely as they should be, right? So some of these relationships may be stronger if sensory conditions are considered in many of the cognitive tests as people get evaluated as we get older. Okay. So the conceptual model that is guiding research in this area currently is the relationship between hearing impairment and cognitive decline. There are multiple hypotheses right now, and they can be summarized as either there is a causal link between hearing impairment and cognitive decline, or it might be some kind of a common factor that gives rise to both of these, or it might be the case that either hearing loss or cognitive decline is present, and one basically provides a second hit to the first. So it might be sort of a combinatorial effect. So there are a number of hypotheses that are generated by these conceptual models, but ultimately it boils down to understanding cellular and brain circuit mechanisms that cause speech and language impairments, following hearing loss. And that's the area that we're really interested in my lab. Like, what is the underlying basis for why hearing loss and hidden hearing loss cause central auditory processing deficits? So what are the changes to central auditory processing with age? So one might say, oh, why not just treat people with hearing loss? In fact, the Lancet Commission report's recommendation was to simply fit people with hearing loss as soon as fit people with hearing aids as soon as the hearing loss is diagnosed, right? But if the statistics on the hearing aids are seen, what you'll see, even in just in the US, only about 15% of the adults older than 50 who have been asked this question and have age-related hearing loss use hearing aids. So it's a very small percent of people who use hearing aids, even though they've been diagnosed with hearing loss. And part of the reason given is that, yes, hearing aids do certainly enhance sound level, but central processing, speech processing is still deteriorated. So hearing aid alone may not be the solution going forward in this kind of a situation. We have to understand what the central, so when I say hearing aids, it's enhancing the sound at the ear. What we also need to do is alter how the sound itself is processed in the brain, and we need to understand what's going on there, right? So what are the mechanisms that lead to temporal processing deficits with aging? So when I say temporal processing, what do I mean, right? So here's a classic study that was published in 2007 by Gordon Salant and others. And here's simply an example of a sentence where somebody's saying, Ruth has discussed the peg. And these are just the acoustic signals that are associated with each of these words. And the gaps here are the gaps between the words. Okay, and this is the natural speech rate. This is the speech rate that is on average pretty natural. And then what they did was they examined 4 groups of human subjects. So they looked at young, normal, and young hearing impaired, and elderly not hearing impaired, and elderly hearing impaired. So they had these different groups. It's a 2x2 design. It's age and hearing impairment. And then they basically expanded different parts of the sentence. So here, for example, the pauses between the words was expanded. Okay, so these little gaps between the words were expanded here. Everything else is normal. Here, just the vowels in the sound, so the oo, ah, I, and the all of those vowels were expanded in this, but everything else remained the same. In this part, they expanded just the consonants, and the vowels and the pauses and all that remained the same. And what they found with this kind of an experiment was that if you, when you compare at the natural speech rate, so this is no TE, stands for no time expansion, and you compare just the young normal with the elderly with hearing impaired, there is a significant loss in detection and recognition of these words. So there was a deficit in speech recognition without any time expansion when you compare young, normally hearing humans with elderly hearing impaired humans. And then when you do the same experiment with the vowels expanded, you see the same trend, right? So there's no benefit in just expanding the vowels. When you expand the pauses, again, there's no benefit. But when you expand the consonants, you see that the hearing and the recognition of the speech becomes relatively the same between, say, young, normal, and hearing impaired elderly. What this is telling us is that there's something about the consonants, the acoustic structure of the consonants, that becomes more difficult to process as we get older. And this has led to the idea that there is something within the consonants that we now recognize as spectral and temporal modulations that become more difficult to process as we get older. So I will use the term spectro-temporal modulation to describe those consonant-like sounds that become more difficult to process as we get older. And one of the ideas going forward is how can we understand why these consonant processing becomes more difficult? How can we improve such consonant processing? And would that increase speech recognition as we get older? And that's part of the idea that we are pursuing. So a major goal in the lab, again, we study mouse models, is to understand, and the advantage of the mouse model compared to studying humans is that we can directly measure from the brain the electrical responses. We can directly measure the cellular changes that happen as the animal is aging, right? And then we can also test hypothetical therapeutic approaches in mouse models which if it's proven to be effective, safe, and efficient, we can then translate that into human clinical use, right? So that's sort of the major goal, is to understand the mechanisms of central processing at cellular and circuit level, and then use mouse to human translation medicine, identify specific biomarkers that are seen in the mouse, and then see if we have the same biomarkers in humans, and then ask if these biomarkers can be recovered, altered, using any sort of therapeutic approaches or behavioral approaches as we go along. So we study two different types of mice, the FVB mice and the C57 mice. These are both your house mice, lab mice, and there are two different strains, genetic strains of these. The reason we study both of these models is because the FVB mouse, as it gets older, does not have that much of a hearing loss. So it ages without hearing loss. Whereas the C57 mouse, as it ages, has severe hearing loss. So we have these comparison, two lines of mice, one which ages with no hearing loss or very mild hearing loss, and the other one that ages with significant hearing loss. So 24, 25 months is basically 2 years, a little bit over than 2 years is the lifespan of a mouse. So these are recordings that were obtained in the auditory system of an FVB mouse that's close to the end of the lifespan, and likewise in the C57. And you can see from these electrical responses, there are strong responses to sounds in the FVB mouse, but not so much of a not much of a response at all in the C57 mouse. So by comparing these two lines of mice as they get older, so if we compare young, middle-aged, and old mice across these two strains, we can ask the question of how does aging alone impact auditory processing, and how does aging combined with hearing loss affect auditory processing? And that's the reason we study these two different groups of mice. So I'm going to talk about two studies. The study one, I'm not going to talk about much. The second study I will start explaining, which is to understand how electrical responses that at baseline with no sound, or when you give a sound and then you measure the responses in the brain, how does that change as the mouse goes from young to middle-aged to old mice? So we're not testing the same mouse as it gets older. So we're not doing a longitudinal study. We're doing cross-sectional studies by using different groups of mice for these different age groups and comparing them at these three ages. And then the third study that we have going on in the lab that I was talking about as the second part of my talk is to ask the question of whether nicotine administration can reverse or decrease some of the age-related changes that we see in old mice. Okay? And the type of electrical recording that we do is known as electroencephalographic recordings, or EEG recordings. And then we obtain these directly from the surface of the brain in two different regions, the auditory cortex, because that is involved in processing acoustic information, sounds. And then we also are interested in the frontal cortex, which is more important in executive function and cognitive function, and so on. So these are mice that are freely moving that we are recording from, and we get signals that look more or less like this. And you can record the same signal from 3-month-old mice and a 20-month-old mice. We can obtain continuous recordings, not necessarily as they get older from 3 to 20 months, but we can get a number of days of recordings from these mice at these age groups. Okay, so here's the first data slide that I will show, which is, it talks about what is known as the decline in the power of gamma oscillations in EEGs as the animal gets old. So this is just a raw trace from a 3-month-old mouse. And this is a raw trace of electrical activity in so this is in the 20-month-old group, and this is in the 3-month-old group. These are two different regions. AC stands for the auditory cortex, and FC stands for frontal cortex. And you can already see, just by qualitative observation of the 3-month examples with the 20-month examples, is that the 20-month example looks a lot smoother in terms of its response, which means that some of the high frequency components that are present in 3 months basically don't show up as strongly in the 20-month-old group. And that's quantified here to the right. And this is what's known as a power spectral density plot, with the frequency of oscillations in the x-axis here, and the power of those frequencies on the y-axis recorded from these signals. And you can see immediately that the frequencies approximately around 30 to 40 to 50 hertz shows a significant decline in power in the 20-month group compared to the 3-month group. This frequency range is what is known as gamma band. And gamma band from a lot of human studies are closely associated with cortical network activity. So how much is the cortex active at rest? And it also indicates function of specific types of cells, neurons that are present in the brain. And we know that decline in those neural populations can lead to a loss of gamma band oscillations. But importantly, for this particular talk, the gamma band oscillations are strongly associated with attention and arousal mechanisms in the brain. And what this suggests is that there is a decrease in the gamma band power, which suggests a couple of things. One, it suggests that the cortical network activity is reduced, and that might be associated with loss of arousal and attention in the older mouse compared to the younger mouse. And it also indicates that we have to go look at specific types of neurons that might be involved in this particular type of deficit. So that's one. The second, this slide shows sort of a similar message, but in terms of acoustic related response. So now this is a response of the brain to a sound that is audible to the animal at both the young and the old age. And when you play the sound and then you record the responses, you'll get a very characteristic response pattern known as the event-related potential, or ERP. And they have a positive going wave and a negative going wave, and these mark specific types of activities in the brain. And what we see is that two components known as the P2 component and the N2 component. So P2 and N2, this is the second positive component, and this is the second negative component of the ERPs, show a decline in older mice compared to younger mice. Okay. And again, the P2 and the N2 mechanisms are thought to be related to attention and arousal mechanisms. So this is in relation to presentation of a sound. And what that again suggests to us is that as the animal is getting older, when a sound is played, they are the arousal and the attention mechanisms that are gathered in the cortex are reduced in the older animal compared to the younger animal. Now, there's a different way to plot the same response. Can be plotted also it's what's known as a frequency time plot. And I mean, without going into too much details, what I want to talk about is this particular component shown here in the warm colors in the orange over here, that is known as, again, a gamma band response. So again, if you see, this is closely related to 40 to 50 to 60 hertz in the response. And this particular gamma band oscillations is also related to attention and arousal mechanisms. And then when you make the same analysis in the young mouse, 3-month-old mouse, and the 20-month-old mouse, and then you subtract the responses of the 3-month from the 20-month-old response, what you see is that there is a decline in the gamma band response in response to a sound. So the blue here tells you that it's less in the old mouse, and the red here indicates that it might be greater in the old mouse. But clearly the gamma band is the region we're focusing on, which is the 40 hertz, 50 hertz region. I previously mentioned that the baseline gamma band is reduced in power. What this is telling us is that the sound evoked gamma band is also reduced in power. So there is a theme that I'm building here, is the loss of oscillation power in the 40 hertz realm, which is thought to be connected with arousal and attention mechanisms. Okay. So again, I'm not going to explain the graphs in a lot of detail, but if you're all interested in that, I can bring it up back in the question and answer session. But the theme that is developing from all of these recordings is that as the animal gets from young age to an old age, there is a decrease in both the baseline gamma power as well as the sound evoked gamma power. And the importance of gamma power is its relationship with attention and arousal. Now, this is a type of sound which is known as the chirp. And the chirp is intended to mimic some of the consonant transitions that we see in speech. Okay, so again, I'm not going to go into way too much detail on the actual data, but what I want to show is that when you present a sound that itself has oscillations, so this is a sound that itself has oscillations, just like the consonants and the speech-related sounds do. What this is allowing us to measure is how much does the brain follow the oscillations, right? So how much is the brain able to keep up with the oscillations that are present in the sound? And the ability of the brain to keep up with the oscillations that are present in the sound is a measure of temporal processing. And we measure something known as phase locking, which is the ability of the brain response to lock to the oscillations that are present in the sound. And we use a measure known as ITPC, or intertrial phase coherence. So the higher the ITPC, so the warmer colors, the higher the ITPC, the better the temporal precision of the response. And the lower the ITPC, the more random the response is in terms of time. So this is a measure that we use to evaluate temporal processing. And again, this is a very complex graph, but basically if you compare the 3-month, which is shown in green, with the 20-month that is shown in black, you can see that across multiple different frequencies, there is a decrease in the temporal precision of the response in the old mouse compared to the young mouse. And you see this in both the auditory cortex as well as the frontal cortex. So what this is giving you is a very precise measure of how much temporal processing deficit is present in the brain of the old animal. Okay? So if you just take those two together, there is a sort of a common theme emerging in our studies of the young versus the old mouse. So resting, sound evoked, gamma power is decreasing with age, as I mentioned. And then the synchronization of the responses, so basically the phase locking ability of the brain to temporal fluctuations in the sound is also decreasing. And sound evoked P2 and the N2 amplitude of the ERP is reduced, again, which is related to arousal. So if you take all of these together, they're all related to potential cortical arousal and attention deficits. Okay. And these are some of the very same deficits that are also seen in humans as we get older. So the number of studies have looked at very similar sort of responses in humans, and many of these are in the same direction, and some cases the same magnitude of change as we go from young age to an old age that we see in the mouse. So what this provides us is with a translational set of biomarkers. So now we have in the mouse a set of electrophysiological biomarkers that mark aging-related changes in the brain. And these markers are translationally relevant because they are the same sort of changes that are also seen in humans. So now we want to ask, can we test potential therapeutic agents to reduce those age-related changes in auditory processing? And we are in the process of developing a number of different sort of approaches. But the approach that I want to talk about today is to the use of nicotine in asking if that can reduce auditory processing deficits. So you might ask, why nicotine, right? So it's well known now that manipulating cholinergic, so nicotine in the brain is a nicotinic receptor in the brain is an endogenous receptor, so naturally occurring receptor in the brain that binds to a neurotransmitter known as acetylcholine. So the acetylcholine is a naturally occurring neurotransmitter that binds to many different types of receptors, one of which is known as nicotinic receptors. And when this binding happens, the neuron's functions can change, right? And this binding between the acetylcholine and nicotinic receptor is important in many different functions, okay? And what we know is manipulating this acetylcholinergic signaling via nicotine administration. So when you give nicotine to an animal, nicotine is the drug that is present in tobacco. And if you provide synthetic or naturally occurring nicotine to the animal or a person, it acts on the endogenously occurring nicotinic receptors and can enhance sensory function. It can enhance reaction time, and importantly, can enhance arousal and attentional mechanisms and cognitive performance. There's a rich literature that shows that this is the case. Activity in the auditory system is also modulated by acetylcholine's nicotinic receptors, and it's modulated at multiple processing levels. And we also know that as we get older, this particular relationship between acetylcholine and nicotinic receptors is impaired in the aging auditory system. Nicotinic receptors decline as we get older, the naturally occurring nicotinic receptors. And so based on these sort of known literature elements, we started asking the question, if you inject nicotine to an old animal and then compare the before injection of nicotine to after injection of nicotine auditory responses, how does the response change, right? So these are some of the same types of biomarkers that I already showed you. So this is in the young group. I showed you that this particular band of frequencies that are shown in red here, known as the gamma band, is a marker of cortical activation and arousal. And I also mentioned to you that comparing young animals to old animals, there is a decline in the old animals. So if you now take an old mouse and give it saline and make the same recording, or give nicotine as an injection and make the same recording, you can see the difference immediately. The nicotine basically enhances the cortical activation and a marker of arousal and attention. So this is nicotine and minus saline. So you're subtracting the saline graph from the nicotine graph, and anything that is shown in red is enhanced by nicotine. And you can see this whole gamma band cortical activation marker is enhanced immediately upon the administration of nicotine. This is in the mouse, 20-month-old. You can also see, and let me go to this one. You're familiar with the chirp response that I told you. This is the type of sound that mimics spectral temporal changes that are present in consonants. And I also mentioned to you, if you compare a 3-month-old mouse with a 20-month-old mouse given saline, the black line here is lower than the green line, which means that old mice are unable to face lock. They show poor temporal processing. But now if you give nicotine, which is shown in pink here, you can see we recover some of the function, right? So it doesn't come back to young levels, but certainly it's better than the old saline treated group. And this is true in both AC and FC. What this tells us is that administration of nicotine improves temporal processing in the old mouse. Okay. So to sort of summarize the data slides that I showed you so far, based on EEG recordings, we know that there is a reduction in gamma band oscillations, right? So that's one of the main findings that we're coming up with these recordings. And there's also a reduced temporal processing or phase locking to these fast spectral temporal modulations that mimic that components that are present in consonant changes. And when you give nicotine to the animal, you can reverse some of these changes. So nicotine increases the power in the gamma band. It also improves phase locking to the chirp stimulus that I showed you. So what this is telling us is that nicotine, however it's working, and we think it's working through the enhanced arousal intention mechanism, what this is telling us is that the circuits, the brain circuits that are processing these sounds in the young animal in a certain way are still present in the old animal. It has to be activated to a greater extent, right? So nicotine may be just one approach in enhancing that activity, and it seems to benefit at least the biomarkers that we are testing. So the good news is that the brain circuits that were present in the young mouse are still present in the old mouse. It's not functioning at the activation level as it is functioning in the young group, but modulators such as nicotine and perhaps many others can enhance the existing circuit to then function like in the young animals. And that's sort of the optimistic view that we're going forward with this research. So at least some age-related brain changes can be reversed with pharmacological approaches. Now, when I give this talk or when I give this poster, many people come and say, oh, smoking, right? So this is not, obviously, this is not saying smoking is a good thing. Smoking itself is associated with all kinds of negative effects, including hearing loss. Smoking itself is known to cause hearing loss. But what this is suggesting is that there are a number of nicotine-like and non-addictive drugs that target cognitive deficits that have been developed quite a bit for Alzheimer's disease, Parkinson's disease. There's been a number of drugs that have been developed that are nicotine-like, but can be taken outside of the scope of smoking, right? So unlike with smoking and tobacco, nicotine can be non-addictive when administered topically or orally. That is when they're absorbed slowly. In fact, we know that is the case because one of the main treatments for smoking, to cease smoking, is to provide nicotine patches, right? So that is a way to tolerate the nicotine's effect with more slow absorption. It prevents addiction and also reduces addiction. So these forms of nicotine are widely available now, and they are inexpensive. Their benefits to central auditory processing, however, in humans are less clear. In mouse, it seems to be providing some benefit in terms of the electrical responses in the brain, but how it benefits in terms of auditory perception and behavior, those are the things that we're testing right now. So nicotine or nicotinic agonists, these are drugs that mimic nicotine, may be profoundly beneficial for aging adults with central auditory processing disruptions, either alone or in combination with behavioral training, hearing aids, and or cochlear implants. And all of these approaches, right? So behavioral training, hearing aids, cochlear implant, and therapeutic approaches are really widely underutilized in terms of auditory processing. And I think that's one of the sort of the segues that I want to give to my colleague, Aaron Seitz, who's going to be presenting next, because he's involved in testing behavioral training approaches. And I think going forward, putting together these two sorts of approaches may be very beneficial to the field. So I want to thank Jeff Rumschlag and Jonathan Lovelace, who are graduate students and postdocs in my lab who did most of the work, and my collaborator, Raju Metharadi, who is an expert on nicotine modulation in the brain at UC Irvine. And my funding sources, thank you to NIH and Hearing Health Foundation, which provides the funding for this kind of research. So I'm happy to take any questions if I have time still.
Thank you so much, Khalil. What we'll actually do is have people drop their questions into the Q&A, and then I'll go ahead and introduce Aaron, and we can have Aaron give his talk, and then we'll move to questions at the end.
Great, thank you.
Thank you so much. So we are joined also tonight by Dr. Aaron Seitz. He's a professor of psychology at UC Riverside. He also holds cooperative appointments in bioengineering, biomedical sciences, computer sciences, the interdisciplinary neuroscience program, and psychiatry. He's also the director of the UCR Brain Game Center. Dr. Seitz received his bachelor's degree in theoretical mathematics from Reed College, a PhD in computational neuroscience from Boston University, and completed postdoctoral work in systems neuroscience at Harvard Medical School. His research is really focused on mechanisms of perception, learning and memory, and multisensory processing. He also, as Khalil mentioned, does translational work. He's interested in using mobile devices in order to read larger and more diverse and traditionally underserved populations. He has a special interest in cognitive diversity, which is broadly defined, as well as in cognitive aging, which is some of the work we'll hear about tonight. And this translational line of work really seeks to create tools that can be used to personalize training based on individual needs to improve cognition. He has over 100 peer-reviewed publications and is multiple millions of dollars in funding, mostly from the National Institute of Health. Tonight, we're going to hear about his work examining or looking at understanding and training to remediate central hearing loss. Take it away.
Now you can hear me, I hope. So thanks for the wonderful introduction and the audience coming tonight, and also for Dr. Razak, who set up a lot of the things that I'm going to be talking about in this talk. And really the focus that I have is looking at how we could understand hearing in the environments that we live in. And so typically we're trying to hear in these, crowded environments. You have multiple people speaking, we have lots of distractions, and that this ends up being very challenging to our auditory systems, especially as we get older. If you look at the typical audiological test environment, you're in this soundproof room, you have headphones on, you're told to listen as hard as you can for these tones that are presented to you. Typically, you raise your hand when you hear the tone. And this deviates in many ways from the ecological settings in which we typically aim to hear. And so the other thing is that we find that the standard audiological test, and Dr. Razak kind of introduced this already, gives you something like this. Is basically what you have is you have different frequencies that you're presented with, some low tones, some high tones. And that if you see on the left, there's this scale ranging from normal hearing to mild hearing loss, eventually to profound hearing loss. And that for each of these frequencies, there's a measurement that gives a range of where you are in the spectrum. And that what you see presented here is that basically the points that are lower, hopefully my cursor shows up, you see, especially for the high frequencies, that these are, in this case, in the blue and the red range, that is suggesting that there's hearing loss. And so it's most of the time, it's the higher frequencies where people show hearing loss, and the lower frequencies, they're more preservation. But essentially what this is showing is just that when at the higher frequencies, the sounds have to be louder for you to be able to hear them. And so on one hand, this is for 80 plus years, the way that we've tried to establish whether somebody's hearing is normal or not. But this concept of hidden hearing loss really reflects the fact that about half the people who self-report that they have hearing difficulties will have a normal audiogram. So they'll do just fine listening to pure tones when they're supposed to concentrate only on those and in these quiet environments. And so if you look at these signs of hitting hearing loss, they include things like hearing people incorrectly, a preference for quiet settings for conversations, feeling easily distracted and unable to focus in noisy settings, a strong sense you have hearing loss even after passing a hearing test. This last one, I think, is really frustrating to individuals. And when we get to, is another slide that I have in common with Dr. Razak, this Lancet study. Here, they're showing a relationship between midlife hearing loss as established by the audiogram, but it's actually underrepresenting the number of people who have hearing loss that may be leading to later life cognitive declines. And so on one hand, it's striking that 9% of this variance of late life dementia is predicted by midlife hearing loss. It's also striking that this is probably a gross underestimate. And so this gets to the work that myself and my group at the Brain Game Center are trying to do, is that we base our approaches off of research like what Dr. Razak showed. So we study neuroscience and psychology. We try to understand how the brain is working. And what we try to do is translate this research into things that we will design new programs and test them out and then disseminate them with the real goal of trying to understand what will predict real life activities, and then also to try to intervene and train them. And so if you look at some of the hearing studies we're working on, and I'll tell you a little bit about some of these, is that we're trying to come up with, similar to the biomarkers that Dr. Razak talked about, where you have these neurobiomarkers. We're looking at behavioral biomarkers. Can we develop tests that will estimate some aspect of your hearing that's more predictive of your hearing in your ecological settings, and use this to be able to better serve hearing healthcare needs? And so we're trying to both develop new tests that are going to be more sensitive, but also try to make them more efficient, because nobody wants to go through 8 hours of hearing tests, which would be the traditional lab approach. Can we get these down to 30 minutes, 15 minutes, 10 minutes to be able to understand somebody's individual hearing? We're also trying to look at how making games and looking at virtual reality can improve these hearing tests. So for instance, take that hearing booth that I showed you before. Well, what if we make it so that you have some of the distractions that you face in a realistic setting? We could do that with games. We could basically, with games, make it so that you have other things going on that are pulling your attention away from the hearing test that you're doing. We could use virtual reality so that we could basically put in some of the multisensory cues. So for instance, to what extent does it make a difference if you're reading somebody's lips? To what extent does it make a difference if sounds actually have place in the world as opposed to be just, presented over headphones? And I'll talk about that a little bit later. And then also, I'll talk a little bit about, we're doing interventions of, as Dr. Razak alluded to, can we do targeted hearing training programs to be able to make it so that at least certain people could train, their hearing, like, a brain workout, to be able to improve some aspect or ameliorate some aspect of the hearing loss. And the other thing is really understanding the relationships between hearing and other aspects of cognition, because that audiogram assumes that hearing doesn't exist beyond the rest of our cognitive processes. But we know that memory and attention and even vision play a big role in our ability to hear things, and hearing plays a big role in those processes as well. And so one of the things that we've developed and we're trying to move forward in research is this application that we call PART. It's Portable Adaptive Rapid Testing. And what this app is doing, and we developed this so that It actually will run, we've done a lot of our testing on an iPad with kind of standard headphones that you could just buy on Amazon or the store. But this also works on a phone or a laptop or a desktop. And we're trying to use this to be able to take hearing tests out of the lab. So can we test people in their own homes? And what this does is both really increase access to hearing tests, but also makes it so that if we need more sessions to be able to measure somebody's hearing over time, or to be able to do cognitive testing as well, that we could do this in the same app and make it convenient for people. And so I'll get back to this in a moment, but I want to talk a little bit about some of the features that we pay attention to in terms of how the brain hears. And so if you think about the audiogram, it's based upon this perspective of auditory cortex and also the inner ear, which what I'm showing you is that, so on the kind of, side of your head, it's kind of highlighted here, is primary auditory cortex. And one of the basic features that people have noticed for years is that it has an organization called tonotopy. It responds in a map where on one side of auditory cortex, you have the low frequencies, on another side, you have the higher frequencies. The thing which is important to realize is that the sounds we listen, and so this is similar to some of the graphs that Dr. Razak showed, is that what we have on the x-axis is time, and the y-axis is frequency. And so these spectrograms are showing you how frequencies unfold over time, the spectral-temporal modulations that Dr. Razak already talked about. And so if you take the word lock versus rock, so these are words that, let's say, native Japanese speakers have difficulty understanding the difference between L's and R's. And this can be understood if you start looking at these spectrograms. If you look at it, we have these little frequency bands, and that, these lower ones are pretty similar between lock and rock. We see this third thing, we call them formants, the pieces that form into phonemes and syllables. We see in lock, it's going down, and rock, it's going up. So I could highlight those. And then the other ones are pretty similar. So what we see is that it makes sense these could be confusable because almost all of the sounds are the same. It's just this third format that's a little bit different. And so for the bias, they might be extracted so that you could kind of look at just this third format. So this is like rock, it's going up. It sounds kind of like this. So this is extended in time. So when Dr. Razak was talking about extending consonants, this is kind of like that concept. And then you could have something like the lock, which is going down. And that one of the things we try to test for is with humans, not just with mice, how are we able to respond to these types of auditory features when they're extracted from speech? And can we predict the extent to which hearing difficulty with speech might be underlined by the ability to recognize these spectral temporal modulations? Another feature that we look at is kind of expressed by the classic cocktail party problem. And so this is similar to the picture I showed you before, but it's even more difficult because now you have a lot of people talking in an inside environment. I think for many of this is like the worst case if you have any level of hearing loss is how can you hear the person you're talking with when you have all these other people talking around you. And that this is typically studied from the perspective of what we call auditory scene analysis. And so the idea is that if you have, let's say, a frog going ribbit, a splash, I'm not going to make all the sounds, and a bird tweeting, that basically if you put them together, that this is a complex waveform. And the question is, how are we able to extract these different sounds? One of the major cues that we use, and there's multiple cues, is sound localization. So the fact that we have two ears allows us to actually figure out where the source of stimuli are. So basically, here's an example of someone with binaural hearing and listening to the difference in how loud the sound is between the two ears, and also timing of arrival of sounds between the two ears, we could tell that all these different sounds are coming from different locations. And what this does actually is it helps us differentiate what these different sounds are. So it's a key aspect of our ability to hear noisy environments. If you have monaural hearing, so you just have, one ear, then, for instance, I think one of the questions was, what happens if you have a tumor? This is something that could lead to monaural hearing. Then all the sounds are on top of each other, and we're much less good at differentiating them. And so kind of the central auditory processing that Dr. Rozak was talking about, an easy way to think about it is that when we look at the ear, it's listening to pure tones. This is what kind of the audiogram is really all about, is how well is the ear functioning. But if we start looking at the brain, it's trying to localize sounds. It's trying to understand these spectral temporal modulations. And that if we really want to understand more about hearing difficulties, or to train them, we need to pay attention to these features the brain is using to hear. And so, for instance, one test that we've developed in part is that we could have you first listen to somebody speaking right in front of you. And they'll say a sentence like, "Ready, Charlie, go to blue 3 now." And your task is just to tap on the blue 3 in that grid that you see on the right. If you're now listening to 3 people, all of them talking at the same time, and so you're supposed to listen to the person that's "Ready, Charlie," but someone else is saying "Ready, Eagle," someone else is saying "Ready, Baron," and you have to kind of listen to the person who said "Charlie." You could do this okay, actually, when they're separated. But if you put all 3 people on top of each other, then this is much harder. And so what we can measure is, and we've done this both on our iPad, at home versus, somebody in a quiet sound booth, is that you see that when the sounds are on top of each other, we call this collocated, you need the target to be much louder than the distractors compared to when they're separated. And we can measure this difference, which we call spatial release. And this is the advantage that if you have this cue that you gain from your spatial localization, enabling you to hear in noisy environments. And the thing which is exciting is that if we test this on our iPad app versus audiological chamber, we basically get almost the exact same results in the same people. And so this shows the feasibility for the mobile hearing tests. Beyond this, what we're doing is that the part also has a whole bunch of different hearing tests. So some are looking at just basic aspects of the spectrum, the standard audiogram, temporal aspects like Dr. Rozak was talking about, spectral temporal aspects, testing for spatial hearing, testing for spatial speech and noise, et cetera, and so on. We're also developing tests of vision. So for instance, the recent study showing that cataract surgery actually gives you a better prognosis of avoiding onset of dementia than if you do not get cataract surgery. Vision is as important as hearing. It's not as well studied in some contexts. And so we have a bunch of hearing tests, we have neuropsych tests, we have tests of things like working memory, your ability to inhibit distractors, task switching, even tests of reasoning. And we're also developing machine learning approaches so that we could basically develop all these tests so that we could make good use of your time when you work with us, so that we could measure a bunch of different features of your vision and your hearing and your cognition, and try to understand how they work together to predict your ability to navigate your daily living situations. Next, of course, is that we want to train hearing. And so building upon the cues I've already talked about is, can we do something that trains both your ability to locate sounds and the spectral temporal modulations and also different frequencies? And so one of the programs we've been using is this game called Listen. Let's say I'll play it briefly. So you have this wisp. So maybe the sound wasn't playing, I didn't hear it, but basically, if the sound is kind of sweeping upwards, you're supposed to move the wisp upwards. And if it's sweeping downwards, you move it downwards. You might have heard that, it's kind of quiet. And so what we do is that as people play this game, we measure their hearing and we adapt the stimuli in a bunch of different ways so that as good as you are, we make it difficult for you. And what we have different levels. And so some are training spectral temporal modulation. Can you hear whether this ripple goes up or down? Some are localization, is the sound left or right? Some are also testing your ability to remember sounds and report whether the next sound is similar or different to the sound you heard. We trained a bunch of college students to start on this. And what we found is their ability to perform speech and competition tasks, so speech and noise tasks, improved from pre-training to mid-training, and then also was maintained a little bit. And so we see that even in college students who are very good hearers, that there's some evidence that we could improve their ability to hear in noisy environments. And then we had a control group that just did a frequency discrimination task. Actually the same game, and we did not see improvements. And so the next step is actually, this was just funded last week, is a new grant to look at what we call mediators and moderators of auditory training. And so our target here is to look for older adults with hearing complaints. And we have a bunch of different interventions. So we have some interventions where we train you on basic speech sounds, like the ones that I showed you. Others where we train you on hearing words, like a lot of traditional occupational therapies do in terms of addressing hearing. We have some programs that are gamified and some not. And so we're basically looking at a number of different ways that people think could be beneficial to address hearing, and trying to compare these to understand them. And the other thing is individual differences. That is our premise, is that not everybody is the same. You're not the same in terms of how you hear, and you're not the same in terms of the types of hearing interventions that might benefit you. And we want to understand these individual differences. And so our aim is actually to run a few thousand people on these training programs and be able to understand these different aspects of cognition and hearing that might predict what types of training might be good or not for you. And ultimately be able to have a better prediction so that when you go to seek hearing healthcare, or you want to address your cognition in general, that we could give you better indications of what things might help you. With this, there's a lot of people who have helped with this, including funding from multiple agencies, NIH. Then I know I'm out of time. I just want to say that you could participate in our studies. So hopefully this link was also sent in the chat. But beyond looking at hearing cognition, we also look at how to train memory, how to train vision. And we have some studies that are focused on just trying to understand these behavioral biomarkers, and others that are focused on how can we train you better. And so we definitely hope that you'll join our kind of community of citizen scientists as we try to figure out these challenges. And with that, thank you very much. Let's see, I need to stop share.
Thank you so much, Dr. Seitz, and again, Dr. Razak, for such wonderful presentations tonight. We have a bunch of questions that have been posed in the Q&A and a couple that I'll try to get to from the chat. I can start going through some of these. If you read through and find any that are easy for you to address in the Q&A, we can also make sure we get to some of them, because I think we only have about 5 or at most 10 minutes to be able to address these questions. Early on in the talk, Marcia Venetsky asked, is everyone with hearing loss recommended to take an MRI to rule out a brain tumor? I don't know who wants to try that one first.
Aaron, I think the recommendation is to go with what your clinician is saying in terms of hearing loss and having to take an MRI for a tumor detection. I don't know if there's any kind of standard recommendation for that or not, but I would follow what the clinician is saying on that one. Yeah,
no, there's not a standard recommendation. There are other indications besides the hearing loss itself that could suggest whether there might be a tumor or not. And so it is definitely best to talk with your own clinician. And actually one thing which is complicated, as like both Dr. Razak and myself are, neuroscientists doing basic research. And so we're trying to develop things that could inform medicine, but we're not really in a position to give medical advice.
Great point, thank you. An anonymous attendee asked, if you or someone you know had a brain injury that increased ringing in the ears and resulted in confusion, disorientation, difficulty understanding speed, difficulty remembering things, and other interpersonal difficulties, what kinds of therapy might be available and how treatable is it?
So
yeah, go ahead.
Go on.
No, after you, Aaron.
Okay, so sadly, this is not a uncommon complaint, and the reality is that the source of these types of problems can vary across individuals, and so some of them can be more inner ear problems, some of them can be more central auditory problems, and some could be related to a brain tumor, others not. And there are quite a number of different therapies that people will try, but kind of once again, this is something where you definitely need to work with the clinician and kind of have them do the set of tests that could help differentiate the possible different causes. And then the frustrating thing is that you might need to get second opinions if the first approaches don't work.
Yeah, part of the difficulty with the type of tinnitus or ringing in the ear research is it's very difficult to develop animal models, because these are sort of perceptions of ringing in the ear, and it's difficult to know if an animal model has that kind of a perception, a phantom ringing perception or not. And that's slowed down the research on developing therapeutic approaches. But there are a number of pharmacological as well as vagal nerve stimulation type approaches that have been developed to treat tinnitus. And again, it's best to discuss with your clinician on precisely what's being clinically tested and what shows promising results.
That actually gets at the second question about, which hearing aids can reduce ringing in the ears. I'll move to Carl Young's question about the power spectra plot for the AC, and that it was kind of confusing. While there was a difference in the gamma band response, there wasn't that much of a difference in the higher frequency band, which I might have thought would be correlated with cognizant recognition, or consonant recognition.
Right. So that's a great question in terms of just trying to understand the basic relationships between the different frequencies in the power spectrum to the types of correlations you might see with the speech recognition. So the gamma band oscillation, which is typically thought of as being restricted between, say, 30 and 60 hertz, is a specific type of oscillation that is elicited, that happens more often when sort of attention and arousal is being aimed at particular type of speech processing issues. Is there a specific correlation between any of these frequency bands with consonant recognition? That is less known, right? It's not very clearly recognized about what is the precise relationship between different frequency bands and different speech processing sort of values. So I wouldn't say I would predict that higher frequency changes might be more predictive of consonant recognition issues. The gamma band is a specific marker of a type of activity in the brain that might be related to basic auditory processing as well as sort of speech recognition issues. So I wouldn't make that link saying higher frequency power in the power spectral density is more related with the consonant processing. That link is unclear at the moment. But the gamma band itself is marking specific things about the brain that might in turn be related to speech recognition and consonant processing.
Great, thank you for that clarification. Linda Crawley asked, how long does the nicotine effect last on enhancing cortical activity in mice?
Yeah, so we've done recordings in mice after acute injections within 30 minutes. And nicotine is metabolized quite rapidly in mice. So within 30 to 40 minutes, the beneficial effects are reduced. But what's interesting is if you have acute instead of acute nicotine, if you have sort of chronic nicotine administration, then the effects are lasting longer, right? So we're talking about effects that last hours to days But we haven't recorded years and so on, or months and so on. So we've done maximum of 2 to 3 weeks, and we see the beneficial effects last that long if you have a chronic sort of administration of nicotine as opposed to acute nicotine. Yeah. And it might be very different in humans because the metabolism can be very different, and the metabolism can also be different at different age groups. So those are all factors that need to be considered. But the optimistic view is that the chronic sort of treatment might be longer lasting. I want to answer a related question that was posted in the chat about nicotine being an addictive drug, right? And that's the point I wanted to mention towards the end of my talk, is nicotine is sort of an approach that tells us that you can actually reinvigorate some of these circuits. So it doesn't have to be nicotine itself. There are a number of drugs that mimic nicotine and may be non-addictive that are already available for Alzheimer's disease and Parkinson's disease, and they've been tested quite strongly. They just haven't been tested for auditory processing. And so, yeah, so if nicotine has that associated addictive issues, either trying the slow release nicotine or nicotine other drugs that mimic nicotine might be an approach to go with.
Great. I might actually skip to the end here. There were a couple of questions for Dr. Seitz. An easy one is, can people who wear hearing aids sign up for your study?
Yes, great. And so the answer is yes, but our typical recommendation is that you do the study without your hearing aids in, partly because we typically use headphones for the studies, and that so we are kind of interested in the idea of what we could do, let's say, training people who are using their hearing aids. And I think that's something that we need to do some customizations, just because headphones would not be appropriate there. But I think that's something right now, if your hearing is maybe not great, but okay, then I think that our training program would be fine without the hearing aids. And then also for the tests, we typically want to understand people's hearing outside the hearing aids context. And also generally what we try to do is that, even if you're not eligible for a study, we often will try to make a version of our programs available so you could try it out, not as a study participant, but, hey, let's see if this, is helpful or not. And so we really try to be responsive as we can to people's needs.
Related to the testing, someone asked about participating in the studies at the Palm Desert campus.
Yeah, so actually, I should have said this, is that our new model is that we will bring our study to you. So we've purchased a few hundred tablet computers, and we have headphones. And so we could basically, we'll sometimes ship them to you and then maybe pick them up at the end. Sometimes we'll deliver them to you. Sometimes if you're part of a community center, we'll try to do a study on site. But yeah, we've definitely learned that it's much better if we bring the study to you than if you have to always come to us. And so almost all of our studies, with the exception of some of the vision ones that require specialized kind of light control, we could do in your own homes.
Great. We probably have time for just one more question. Dr. Razak, if you want to look through the Q&A, if there's any questions that you can answer, I would suggest maybe you fill them in. I think I'll do one more here for, where was it? Actually, there's one that was interesting. Could a patient use an AI model of a single person, such as a husband, to better hear that person?
So I'm definitely interested in the context of that. But one thing which is really intriguing with some of the modern hearing aids, and in fact, some of them are even sold to people without hearing difficulties under the name hearables, is that they're trying to use AIs to intelligently amplify the sounds you want to hear, and downplay the sounds that you don't. And so it's not exactly the same as creating an AI of your husband, as much as basically trying to understand what are the frequencies that are informative in your husband's voice and making those louder and other things quieter so that you could listen to him better. And it's pretty impressive the extent to which hearing aids have been developing in the last few years. Some of them actually try to, measure brain activity to detect where they think you're attending. So they'll amplify things directionally based upon where they think you're attending. Some of those worry me because if you sneeze, then it gets brain waves that it doesn't know what to do with, and it amplifies who knows what, but they're working on it.
The last question I'll put to both of you, and perhaps then Dr. Seitz, you could also spend a minute to see if there's anything else that you can answer in the Q&A. But the last question I think we should leave on is if there are any ways to prevent or mitigate decline. I believe they mean hearing loss and then in turn the cognitive decline that we would see.
Yeah, so we definitely know noise exacerbates age-related hearing loss. So one of the things that really worries me studying in this field is the really enhanced use of recreational in-ear devices and over-the-ear devices that have just been dominating in the last 10, 15 years. You see a lot of children and kids and adults wearing earphones and headphones all the time. A lot of times it's not just how loud the sound is that can cause noise-induced hearing loss, but also the duration of those sounds that go into the ear. So certainly mitigating, reducing noise exposure, including recreational in-ear devices is one way to reduce age-related hearing loss. And that was certainly a recommendation going forward.
Yeah, and I'll just add that, my general advice is do everything good. So, people say that exercise is important. You want to get some exercise. People say that better diets are better. You want to make sure that you have a good diet. People say that you need to have the right type of cognitive stimulation. So I think that apps like the ones that I developed could be part of that solution, but generally, since we don't know what it means to be cognitive stimulating, what you want to do is to do many activities that will stimulate different aspects of your hearing, your vision, your memory. And so some of the advice, is, learn a new thing every day. If that's too much, well, pick up a new skill, every few months. And so basically, and when you're good at it, you're not being challenged. And so you're probably not learning as much. And so you want to pick up something else that challenges you. And the more that, you're, challenging your body with physical fitness and challenging your senses and your brain with different types of stimulation and activity, all these things seem to add up. And that if you just try one solution, it's probably not going to do all what you want. There's also pharmacological solutions for some things, but all of the above solution seems to be the best one at our current state of knowledge.
And if I may add, if there's one thing that's sort of a takeaway from both of these talks is that if there is any sense of hearing loss at any given age, don't delay getting it looked at.
That's a great message. Thank you again so much to Dr. Razak, Dr. Seitz. A pleasure hearing your talks and moderating this Q&A. Wonderful answers. And thank you to everyone for tuning in with us tonight.